Okay, hi everyone. How are you today? So today, the last part of this chapter, which is about mutations. So we are going to take a look at brief principles, how we, in several different ways of getting mutation, and three examples of some interesting mutations as a final examples of this chapter that we are going to take a look at. So first off, the definition of a mutation. Several times I have introduced this concept. So DNA sequence changes. Some people ask that, what about the RNA sequence change? Will it be also counted as a mutation? Yes, it does. However, the sequence change in the RNA, yeah, that also happens. The DNA sequence change is not the only one occasion that you can have an alteration during the whole process of a cell. However, when the RNA sequence changes, it usually doesn't matter because usually the amount of RNA, the same sequence of the RNA that is to be produced is a huge, so many multiple copies of the RNAs are produced at a time in a given time, and the the likelihood of having a specific RNA being altered out of tens of thousands of multiple copies of RNA is very slim. So you're not going to get affected. The chance of getting actually affected by this sequence change of this RNA, individual RNA, is negligible. So usually you don't talk about the RNA mutation, usually. So several different types of mutation we can think of like this. Namely, substitution, deletion, insertion. If you think about these, what they are, substitution, switching. Right? So if you want to have an absentee, if you want to skip the class, then instead of you just call your friend, ask your friend, and then your friend can come in place of you is basically the substitution. So no one will notice if that is the case usually. On the other hand, deletion is simply you don't care about, simply don't come to the class. So you're missing, actually. Your position is empty, absent. Insertion, on the other hand, you're not supposed to come here, but somehow come here as an additional one. We can define as an insertion. So these three mutations can happen at one single nucleotide level. Then we call that as a point mutation. So all these three mutations, different kind of mutation, can happen at the level of a single nucleotide. Then we call it as a point mutation. Single nucleotide level. What I'm saying is, not only can this mutation occur at the single nucleotide level so that we recognize as a pain mutation, but also all these three different kind of mutation can occur at a larger scale, like a whole chunk of the DNA segment can undergo this type of all three different types of mutation. Okay, so this three things can also occur at a very minimal scale of a point mutation level of single nucleotide level, but also can occur as a larger scale. Sometimes actually this thing can happen as a huge chunk of a large fragment on the chromosomal level that we are going to take a look at briefly today too. So substitution, deletion, and also insertion can also happen like that. And if that happened, if this was part of the messenger RNA, the protein coding region of the DNA, then you have some serious problem potentially. If this thing happens on the non-coding area, non-protein coding area, usually the effects will be negligible or no effect at all occur or very minimal. However, if this thing happens on the area where actually it will be converted into the information that is needed for protein coding, i.e. the messenger RNA region, then actually the identity of individual, this amino acid can be altered because of that. Then you have some potentially a big problem. So as you can see, base substitution, for example, here, one amino acid, originally it was a G, has been changed into G, and then as a result, changed into A, then as a result, this GGG, the codon, 3-nucleotide codon, GGG, supposed to code for glycine, the amino acid. Now it has changed into something else, which is a serine, which was not meant to originally. Then it could actually affect the function, original function of this whatever, the mutant, the protein function too. And also, by deleting one nucleotide, you can have totally different types of amino acids afterwards, after this deletion point, because a lot of times this point mutation of especially deletion or insertion of one or two nucleotides is especially the problematic. It will definitely result in something we call as a frame shift mutation, one or two nucleotide level. If that happens at the 3-nucleotide level at the same time, in a given time, then the effect, the consequence would be much less, potentially much less dangerous. However, if that happens 1 or 2 nucleotide level, then because of this, the nature of this coding, which the codon consists of a 3-nucleotide, right? Then you have the disrupted order of this codon. Then we recognize that as a frame shift. What is frame shift? Some shift of the reading frame. What is a reading frame? This is an example of illustrating what the reading frame is here. If you try to read the sentence, interestingly, this sentence consists of all 3-letter vocabulary. 3 alphabet. What a coincidence. And then if you read, the red dog ate the cat, that makes perfect sense. It is making up one perfect, completed reading frame. Now then, something happened, and then you try to read this. Does it make sense? You try to read. The got that hecka doesn't make any sense. What happened? Can you identify the problem? What happened? Yeah, this E has been deleted out because this sentence is still maintaining the 3-letter reading frame. So the E was out, so then automatically they just filled in whatever the 2 following alphabet to form once again the 3 reading frame, this rule. That's why all of a sudden this sentence has become totally out of sense. That's exactly what's happening. If you delete one nucleotide, then from that point after, all these following reading frame will be disrupted and totally not making any sense in terms of if three of those alphabet was codon, so that each of these three alphabet combination was something meaning some specific amino acid. Now what? All of these three codon things is totally non-original, out of order things because of this single point mutation, the deletion point mutation. Now here, what happened? The red dog ate the cat, and then also totally, it's unreadable even. What happened? This time, something was inserted. Single nucleotide or single alphabet was inserted and then disrupted the following reading frame. Then that's exactly happening in the case of this, your messenger RNA coding area, the gene. If deletion of one nucleotide or insertion of one nucleotide, because this reading frame is maintained in a 3 nucleotide codon basis. 1 or 2, single nucleotide or 2 nucleotides of deletion or insertion will always cause this type of a problem. So as a consequence, you may have some totally irrelevant amino acid may come, or in some occasions, you may not even have any protein produced if that happens in the early part of this messenger RNA, and then immediately there are 3 chances of having that one of those 3 stop codons can inadvertently result in. Then the translation will immediately stop right after the mutation point. Then no protein whatsoever will be generated. So as a result of this frame shift mutation, either you have a missense mutation or a nonsense mutation can occur. Missense mutation, in missense mutation, you still have some protein product of totally different amino acid composition. So wrong protein. In missense mutation, wrong protein, incorrect protein will be generated, which is a garbage, which may actually be harmful in your well-being, welfare of your cellular process. In nonsense mutation, no protein will be produced. Why? Because of a premature stop codon. So like, for example, missense mutation, as a result of this deletion, still translation will be gone, but originally you intended, but incorrect amino acids are generated. And in this case, we call it as a missense mutation. Incorrect protein is made. But more seriously, I don't know actually. I'm not really sure whether nonsense would be more serious or missense would be more serious, because if you're not going to be able to produce your intended correct protein, sometimes actually you better actually not even produce no protein at all, rather than producing totally incorrect garbage protein sometimes. So anyway, nonsense mutation, the reason why nonsense mutation is nonsense mutation is result is because sometimes as a result of this frame shift, you may have this premature stop codon is generated, for example, like insertion of, in this occasion, particular example, as a result of this U insertion, then simply UAA consists of one of the three stop codon, right? Then, just right after making one methionine amino acid, this whole gene, no matter how long this actual original messenger RNA sequence, not get to produce any protein at all because of this stop. Is called nonsense mutation. On the other hand, deletion of three nucleotides sometimes actually has a least serious effect, consequences, because you still preserve the reading frame. Let's go back to the reading frame thing over here. So let's say here, a deletion has occurred. So RED, 3 letter, it's been entirely deleted out. You try to read. The dog ate the cat. Still, the red has been out, but still it makes sense. The total sentences, this makes sense. So probably the function-wise, the protein's function, you lost three nucleotide. As a consequence of the losing three nucleotide, you may have lost one amino acid out of entire your protein. But in many cases, chances are that protein may still function somehow more or less normally. So this, once again, why? Because this codon is being maintained as a three nucleotide thing. So anything changed, any change occurring at the three nucleotide level, like threes are missing at one time, or threes are inserted in one time, are usually, at least you don't get to disrupt the whole reading frame. So in most of the cases, the consequence impact would be minimal in that case. On the other hand, if that happens at the level of one or two nucleotide basis, then the impact would be usually very serious, because it will change the entire, or either through this missense mutation or nonsense mutation. In any occasion, it will be just like that. But sometimes you also get a lucky break, especially in the case of substitution. Not a deletion or insertion, but substitution. I'm not saying all the substitutions will be harmless, but some occasions this base substitution may result in some change in amino acid, but in some occasions it may not even result in any change in the amino acid identity, because first off, our codon system is redundant. Several different codons code one single amino acid. If you go back and then look up, examine the codon table, 3 or 4 different combination of codons are all pointing to one same amino acid. So like, for example, if this single nucleotide has been changed into, but fortunately the change was something in the range within still the category of the same codon, because codons are redundant, then consequence is none. You still get to have the same identical amino acid of the originally intended. On some other occasions, even if you have this change due to this substitution, actually the consequence is a change in the amino acid. However, still you don't have any effects, because you have 20 different amino acids, but some of those amino acids can be used interchangeably because the chemical nature is very similar. Then you're very lucky. So due to this mutation, this original amino acid has been changed, replaced with something else, but these two amino acids are actually basically replaceable because of this very similar chemical and biochemical nature. But ask a Siri? I don't want to ask you. So these two occasions we call as a silent mutation. Even if mutation occurs, then the effect can go unnoticed, either one of these two reasons, then we call it a silent mutation, as opposed to missense or nonsense mutation. So the consequence of a mutation, we can list three different locations, like as a result of mutation, you can have either missense or nonsense, or in some occasions, there's silent mutation. But you should be able to distinguish, like nonsense and missense mutation usually are caused by what? Insertion or deletion. Insertion or deletion of one or two nucleotide. But this silent mutation always can only be possible by substitution. If you think about it, substitution, right? Practically speaking. So some examples of different type silent or missense mutation can arise as or summarized in this figure. You can check that bit later. Now, as I said, this mutation, three different types of mutation can occur on a whole larger scale of a chromosome level. Chromosomal scale level, you have one more kind of, at the chromosomal level, not only this deletion, duplication is something in, so back up once again, totally. At the huge chromosomal level, we can view this mutation in different ways, so that usually four different types of chromosomal level or chromosomal scale mutation are possible. They are, one is deletion. You can easily understand what's deletion. Huge chunk of whatever on certain area of a chromosome, this huge chunk of chromosomal DNAs are totally missing for various reasons. And on some occasion, duplication, huge area is being copied and by mistake, so that additionally this is repeated, and we call that as a duplication. Inversion is this orientation of a certain area is being flipped upside down. So inversion also occurs. And translocation is simply whole chunk of one segment on one chromosome is being moved out to the other chromosomes. That is the most serious impact. This translocation has a really, really serious consequences. But figuratively, if you try to imagine what's deletion on the chromosomal level, for example, A, B, C, D, E, F, G, H, this whole area is represent one single chromosome, and this segment D is entirely missing. Okay, duplication, the 2BC area is repeated once again, so that you have additional copy of the entire region, is what we call as a duplication. Inversion, on the other hand, as you look at it, A, B, C, D, E in this order, chromosomal segment has been maintained, but somehow being flipped. So now A is just followed by D, C, B in this order, is what we call as inversion. You may think, inversion, what's the big deal of this inversion? You didn't lose anything, and you didn't have any additional thing, and still does it matter? Yes, it does. Think of why. Why still this BCD, like this inversion, seems like just harmless, but in many occasions, this inversion will also cause a big problem. Because of what? Can you guess? You always have to think that what? Why do we have this chromosome? What's the function of this chromosome? It's a container of our gene. And why do we have our genes for? For what? Gene expression. We have to have some gene expression. And gene expression always have some kind of order and plan. This entire plan, when to express certain gene, and how much we are going to express certain area of gene is totally disrupted. And remember, the simple gene expression program's disruption may even result in such a serious consequences like disease like progeria. Yeah, so we cannot really think this as a simple lightly. Translocation on the other hand, here you see two different chromosomes. And one segment of this large section of one chromosome is being moved onto the other chromosome, and this, in this case, is like we call it reciprocal translocation. So now then, the other chromosome's part is also moved onto the other chromosome, and sometimes you don't even have this kind of a reciprocal translocation, but simply one section has just simply moved to the other, and that's it. And this translocation is often observed in cancer cell. Whether it is as a translocation, it causes cancer, or as a cancer develop progress and all kind of regulation mechanism is somehow messed up. So more frequently this translocation occur. We don't really exactly point out which one is which, but anyway, this translocation of the large scale translocation is always a very good hallmark of cancer cells. Sometimes, because of this simple translocation, some of the genes is unusually activated and cause cancers. Some other times, because of this whole total lost control over regulation, actually accelerate this type of mistake too. But whatever it is, this often frequent translocation is not usually observed in so-called normal cells, because if that occurs, this normal cell cannot function. But cancer cell, still with this type of total disruption, cancer cells somehow still stay alive. That's what actually cancer cells for. So you can get to see a lot of these things. Now, another very interesting question that I'd like to ask you is, among these four different types of large-scale chromosomal mutation, which do you think, if ever, which of these four would be actually most beneficial? Don't get me wrong, all these four different types of mutations are really, really bad, deleterious. If you ever have this one of these four, then chances are you're not going to be able to survive you're not going to be able to live a very healthy life, to put it lightly. Okay. But still, there are a lot of different occasions, so you may still have some of very lucky break. So having said that, out of these four, which one of these four would have actually the best chance of providing some contribution to actually evolution? So at the flip side, one of these four different mutation has actually contributed so much in progress of evolution, actually. Which one do you think would happen? played such strange role. This may be actually a very good topic for your upcoming final exam question. A, the deletion. Why? You mean the insertion? The A is deletion. Deletion is missing. So you wanna rephrase? Answer is duplication. Duplication has contributed very significantly to the process of evolution. Of course, I'm not saying all the duplication has done such thing. Probably most of the duplication probably has affected so negatively. But some occasion, very, very rare occasion, then this duplication, thanks to this duplication, or this organism could have evolved into something more advanced, something new, having new traits. Why? Exactly. Thank you. Exactly. So when you have only single gene, one copy, if this gene gets mutated, usually the chances are the concept of mutation is you're dead because you will lose the function of the original function of this gene as a consequence of the mutation, or because mutation by nature is just change in DNA sequence. So leading to change in the amino acid sequence of the protein that is produced from the gene. And what are the chances by this alteration? What is the likelihood of this mutated version of protein act normally or something a better function? Extremely, almost none, almost. I'm not saying there is none, but almost. So the result of the mutation usually always is such a very bad consequences. But the consequence of duplication, you get another copy. Now you can play around this experiment. The mutation will always occur randomly. So the chances are mutation occurring on both of these genes will be extremely low. So usually mutation will occur another copy of your gene, but still retaining the original copy of your gene functioning normally. So that in this case, you have so much higher degree of freedom. You can play around. So you're not going to get killed. Just because your gene is mutated, you're not going to get killed. And some occasions, very, very rare, some lucky occasions, maybe you won the lottery, then this gene, mutated gene, may actually provide new trait, a better, superior trait. Actually, one very good example is hemoglobin. Usually there was only one single copy of a globin gene, but hemoglobin protein gene. But through this duplication, several different copies arose, and slightly different version of this hemoglobin protein could change so that each different version can provide some slightly different roles. And there are so many other such examples of obtaining a new different function of the same gene through this duplication has been observed and discovered throughout so many different organisms. Now, So, having said that, we are going to take a look at three different actual real example of the mutation. The first one is the famous sickle cell anemia. Technically, this sickle cell anemia is caused by a base substitution mutation. One single nucleotide has been changed into something else differently. And the mutation affected this one of the hemoglobin chain protein. There are two different kinds of hemoglobin protein, and they are all combined to form one fine functional protein shaped like this. This is a model, protein structure model of hemoglobin protein. So anyway, so we call it as there is an alpha chain and beta chain, two different hemoglobin protein. They are combined together to form one functional unit of hemoglobin. And mutation, single base substitution mutation occurred in beta chain at the early part of this beta chain DNA sequence. As a result, so nucleotide A has been changed into T, AT substitution. And as a result, actually amino acid has been changed. E6V is one kind of amino acid, glutamic acid E, has been changed into valine, and the position of this amino acid was the six amino acids from the start of the big protein, which is a 200 or 300 amino acid long protein. So at early stage, one single nucleotide change actually result in the amino acid identity change. So this time, it didn't have, unfortunately, it did not have any silent mutation. But luckily, because of this substitution, it didn't have any frame shift mutation, so it didn't have any entire large-scale missense mutation, nor any nonsense mutation. So it seems to be very minimal. You still have a protein produced, and the amino acid change is just one amino acid out of several hundred students. Hey, let's say in this classroom, let's say there are several hundred students were there, and one student didn't come, and instead of her, a friend came in. How am I supposed to notice any difference? The class will just go on without having any adverse effect or disruptions. But this time, because of this one single amino acid substitution, it actually has made a big difference. And that's why the person having this mutation had to suffer this disease called the sickle cell anemia, which is very serious multiple effect disease. Why? Because amino acids, glutamic acid is quite different in chemical nature from valine. These two amino acids this is really unfortunate mutation. A to T transition or substitution will always occur. And a lot of times this will not have any effect. But very unfortunate event of this time around, it made such a big difference because of this amino acids chemical different nature. So why then? Having this one single amino acids difference, the protein made as a result, the mutant hemoglobin protein, instead of having this original ball-like shape, we call it as a globular shape, they form piled up accumulated version of hemoglobin protein, which is non-functional, this hemoglobin protein. The red blood cell at maturity all a single red blood cell has is a whole bunch of a pile of hemoglobin protein. And if those hemoglobin protein is a totally different structure of hemoglobin protein, then actual shape of this red blood cell will also change. So the actual shape look like sickled shape. That's why we call it sickle cell disease, sickle cell anemia, because if you don't have any functional hemoglobin, then you suffer from anemia. Is like a lack of red blood cell, but in this occasion, you still have a red blood cell, but those red blood cells are not functioning because their function is carrying the oxygen. They cannot provide this oxygen delivery. That's why you also have the same effect as if you are having the anemia. That's why it's called the sickle cell anemia. So normal hemoglobin molecules appear like this, single individual hemoglobin protein, each of which functioning in carrying several molecules of oxygen. But this sickle cell anemia patient, hemoglobin proteins clumped together like. And these, with this form, with this fiber form, they cannot function. They will simply settle at the bottom of the red blood cell and pile up. That's why round, cutie, nicely round shape of red blood cell will become this crescendo moon-like sickle shape because of this. And actually, the reason why this hemoglobin protein, mutant hemoglobin protein in sickle cell anemia disease have this type of a clumping is very interesting and also very telling. And this is the mechanism. Normal hemoglobin protein have this kind of shape. Now, why the sickle cell hemoglobin have this aggregation or clumping is because this glutamic acid is hydrophilic. You don't have to know this, but glutamic acid chemically is hydrophilic. And in protein overall structure, hydrophilic proteins are exposed on the surface because hydrophobic part is water hating, so they always try to fold inside naturally. That's how you generate automatically any given protein structure. And this hydrophilic amino acid has been substituted into hydrophobic. Valine is hydrophobic. And all of a sudden, this hydrophobic protein is now at the position of being exposed on the surface. And then it's a big chaos. This protein, hemoglobin protein having hydrophobic part exposed to the water is being panicked. So what their best way to compensate such disaster is try to hide. So let's say this is a hydrophobic part exposed due to this mutation. But the only way they can minimize this disaster is clumping together so that all these hydrophobic part can be exposed, just like hydrophobic molecule tend to cling together to minimize the water exposure. That's what's exactly happening in this occasion. And that is the reason why this originally individual ball-like hemoglobin protein now is forming long clumped aggregates. This protein, which is really disastrous to the function of this red blood cell. Okay. Now, the reason why I'm saying this is something telling is, this is exactly also happening in Alzheimer's and diabetes and Parkinson's. Same, this principle of normal protein is being changed into something different so that some part become hydrophobic so that inviting these aggregates of this protein is also observed, being the same thing is observed. Even if that occasion it was not about this hemoglobin protein, but some different protein due to some different type of mutation, that particular protein, normally, that's a normal cellular protein, is now become aggregates and causing, and even the mad cow disease is also type of such the malfunctioning protein is due to the difference changes protein function exposing this hydrophobic part become this aggregate and then this destruction of the actual cell. It's all the same principles is being observed in many major human disease is something that is telling. So think about it. If you have a problem in delivering oxygen, then it's not something only about the oxygen transport anymore. All the many different important metabolic process that is being also dependent upon this oxygen supply will also be severely affected. And obviously it is so natural that this one will have so many different multiple effects. Okay. And genetically speaking, this type of one mutation is affecting so many, causing so many different type of trait is called pleiotrophic effect. It's from the viewpoint of simple classical genetics. But if you think about the nature of this mutation from the molecular genetics viewpoint, it is so obvious. Of course, you will have. Because of this nature of this mutation, there is no other way around that you will have so many different ways to cause these different phenotypes, of course. So it's nothing new. So like so many different results. So if this was something, so there are so many different examples of this type of single gene mutation will have multiple different traits. We collectively, genetically speaking, we call that as a pleiotrophy. Okay. The other way around, single character, single trait is being formed, being controlled by many different genes. We call it polygenic inheritance. There are so many different examples of like the characters or traits determined by polygenes, including human skin, human height, human intelligence, even eye color, you name it. Most of these traits are actually the result of interactions of many different genes. It's very rare actually. Single gene is responsible for single trait. It's not really widespread, but that's something that rare. So, could you correctly guess that this sickle cell anemia be recessive mutation or dominant mutation? It is recessive mutation, but could you guess if you, just by hearing, okay, this is the nature of this mutation, were you able to correctly anticipate, oh, then this gotta be a recessive mutation? Remember, the key to determining whether this gene is this mutation is going to act as a dominant or recessive is whether the functionality, whether as a result of this mutation, the function of this protein is going to be lost, then it's gotta be a recessive mutation. This sickle cell anemia, the clumping, so you're not going to be able to, this mutant carrying, the cell carrying this mutant will produce non-functioning protein. However, if you had normal, so to speak, normal protein, hemoglobin protein, still produced this normal protein still should be able to provide the role of carrying the oxygen. In this case, yeah, you have still some garbage which is not going to be able to do any function, but still you have functioning copy. So carrying oxygen, transporting oxygen shouldn't be affected, like largely speaking. Yeah, that's the reason why the sickle cell anemia, the mutation is a recessive gene, right? So only you have both copy of this beta hemoglobin gene have the same mutation, then you're not going to be able to actually live because you don't have any functioning hemoglobin to carry this oxygen. But other than that, the heterozygous should be relatively normal. I mean, unless you decide to live in like Himalaya mountain or places like that, where oxygen is really, really scarce on the normal, usual habitat for human, you're not going to have any big impact. However, this heterozygous have an unusual advantage. So, like if you think this heterozygous carrier, then the chances are throughout the maintenance of a population, by chance, some individual will somehow, unfortunately, will become homozygous for this mutation. Then this individual will have a serious problem in survival. So they will be eliminated sooner than later, eliminated out of this population. So in this way, this allele, this mutation, the gene allele, will be gradually eliminated out of this population, right? That's usually what you can expect. However, especially in Africa, this frequency of this sickle cell anemia mutant allele is unusually high because of this reason. These heterozygotes have some selective advantage against malaria infection. So if you have, there is a slight, when you, as a cell, the red blood cell, then you get to produce more carbon monoxide in the red blood cell. And surprisingly, this carbon monoxide is very effective in preventing the malaria parasite infection. There is a more in-depth, detailed story about why this malaria is so harmful for us, but yeah, I will just shut up. Because this parasite is actually feeding only the hemoglobin protein for some reason. Among all these different nutrients inside of the human, they only love and they just feed only on this hemoglobin protein, and that's the reason. But anyway, so because of that, this heterozygous has some advantage. And if you look up the area of this malaria, the heavy occurrence area of malaria and the frequency of the sickle cell mutant, so really nicely, perfectly overlap, which indicate that because of that, the frequency, this region, this area, this mutant gene frequency is so high. The second example is a cystic fibrosis. Now, this time, this cystic fibrosis mutation is caused by 3 nucleotide deletion at a time. Now, you don't have the reading frame change, luckily here. 1 or 2 nucleotide deletion, it would have definitely caused frame shift, but the 3 nucleotide is entirely at once deleted. So you only miss 1 amino acid. And usually, if that was the case, usually, as you saw in the example of the red dog ate the cat or whatever, one single deletion of a 3 letter doesn't really affect any overall meaning. However, in this occasion, it's very, very unfortunate 3 nucleotide deletion, so it has a very serious consequences. This mutation is regarding in this particular protein named the CFTR. The full name is whatever, and this is a transporter protein, the ion transporter protein present on our cell membrane. So by convention, we describe this mutation as is like this: delta, Greek letter delta, is synonymous to D. Okay, D stands for deletion. So deleted. Delta F508. F here, this F means particular amino acid, phenylalanine. The phenylalanine at the position of 508. This is a huge protein. Really, really, really big protein. And so the position 508 of this protein is still rather regarded as considered as an early part of this entire protein. And this single amino acid is simply deleted out. And chances are, if you think like reasonably, this type of mutation should not even affect at all about, because for one, protein is so huge. Out of such a huge protein, maybe 1,000 of amino acids maybe, and then one amino acid is missing, what kind of an effect would they have? None. But in this occasion, it has a very, very serious effect. And the normal function of the CFTR, as I said, is an ion transporter. Notably, a chloride ion is being transported, and in return, through this transporter process, cell maintain constant flow of liquid outside of our tissues. For example, our this air tract or some other area where like liquid is being flowed, especially the function of the CFTR protein becomes so important. The cartoonic version of the CFTR protein, which you can ignore, whatever. And this is big protein, and embedded in, due to this transmembrane channel protein is being embedded, but I'm not going to ask anything about this type of thing on the exam, so you can totally ignore. This is simply showing how the overall structure of this protein. That's all. Now, why only this single amino acid deletion is such a big deal? Because of this mutation, protein folding is being affected. Remember, this protein is a membrane protein. So where do you think the place of synthesis for this protein would be? Where, if you remember still? Is there any crazy enough students still remember this? The place of synthesis for this type of a membrane protein would be where? The surface of rough endoplasmic reticulum, right? Remember? And so this protein, after finishing the translation, this protein will be pushed inside of this rough endoplasmic reticulum, inside of which two very important process will be carried out, which is, one is protein folding, the other one is glycosylation. Yeah, thank God these two things are like that. This is not a part of your final exam content, thank God. But anyway, that's what's going to happen. So protein folding inside of this ER, protein folding correctly should be done. But due to the missing of this phenylalanine, particular phenylalanine amino acid, this protein cannot fold correctly, incorrectly fold. And those incorrectly folded protein will never be able to escape this ER. They will not allow. They give 2 or 3 different opportunity to correctly fold, and if still this protein cannot fold, if it turns out that this protein is incapable of correctly folding, and this protein is actively degraded into individual amino acids inside of this ER. So if you were carrying, luckily, what do you think? Would this mutation be dominant or recessive? Tell me. Recessive. This mutation would be recessive. So if, let's say, if you have normal gene and mutant gene of this CFTR, then normal gene product, which will be able to correctly fold and will be able to escape the ER and then go their usual place of the cell membrane. So should be able to provide the normal function of this transport. Yeah. However, because this is simply non-functioning one. However, if you are homozygous for this mutation, then the cell membrane of this individual will not have any single this CFTR protein because they are all degraded inside of this ER. None. Then now you have a big problem. So, like on the surface of this our epithelia, thanks to the function of this ion transport protein, so normal water flow will be maintained. But if you can provide this function, then no water, just like the river, which is incapable of providing a decent water flow, then we'll have such a lot of cellular sediment, include inviting so many different micro, like the bacteria, okay, stuck into this area. So thick mucus containing so many different infectious agents will result. And because of that, you became so much vulnerable to many different types of infections. So this is going to be another such a pleiotropic disease. So many different tissues and cells function will be affected by that. So that's what it is about this. Yes? Because of its bacteria, so you have to treat with antibiotics. Yes. But if that's like superbacteria, then you have no rescue. And this is recessive mutation, and actually the only reason why you have to learn this disease is because this is very rare disease, genetically speaking. It doesn't really occur. But for some very weird reason, this Northern European heritage population, so like white people, has such very high, unusually high frequencies, even 1 in 3,000 homozygous phenotypically, like affecting this disease, which is very high frequency due to their particular lineage, their ancestral lineage, or for the white population. So those white people, this is a very serious genetic disease. But for black and Asian, who cares? Because you never actually, the chances for you discover this type of rare disease is extremely, extremely slim. But unfortunately, all these textbooks were written by those white people. So that's why in every different biology textbook, you are forced to learn this cystic fibrosis. But anyway, for our own sake, this is something actually we can learn a lot about, because this is unlike the general such occasions like a 3-nucleotide deletion or insertion, which would not disrupt any reading frame. In this occasion, yes, of course, it didn't disrupt any reading frame, but even so, you have such a big problem. Why in this occasion? Protein structure, protein folding was like the reason and the importance why the protein folding is so critical. Why? Because an incorrectly folded protein is not able to provide any function is the reason we can learn out of this occasion too. The last example is lactase persistence, my personal favorite, because I cannot live without ice cream, and even at this age. Even last Saturday and even Sunday morning, even watching the football, college football in the U.S., I emptied the whole big ice cream carton myself during that time. I had a very good time, enjoyed with Sookmyung too. But anyway, so so that's why I actually personally became interested in this particular mutation, in that it is a base substitution mutation. So this mutation actually allows you to be able to still keep digesting this dairy product. Without this mutation, you are programmed to lose your ability to digest this dairy milk product as you grow older. Because guess what? The milk product, you only need the ability to digest this milk product when you are a baby, when you are dependent upon your mother's milk. Afterwards, you are going to have different source of food, the adult. So humans are programmed like you're supposed to lose this ability. When the enzyme, the lactase, the name of this lactase is responsible for digesting all those milk products, including ice cream and all different yogurt and any different kind of milk dairy fruit product. And the expression of the gene, this lactase gene, will dwindle as you mature individually. So like most of the Asians actually, it's like that. So that is the reason why when you are a child, it's just totally okay, but when you become 40 years old or 50 years old, something, then just like my wife, when we were 20s, we both together, we were able to enjoy such ice cream overnight, but not anymore. I can only, it's me only now. She always wants having this dairy product, her stomach become difficult to digest. A lot of gas build up, right? Because inability to digest these things, so your microorganism inside of your gut will take the place to digest this thing, and as a byproduct, a lot of uncomfortable gas will build up, and that's why you. So it is not such a serious problem. You will live with this. But anyway, mutation allows. So some people carrying this mutation will have no problem of digesting this even at the age of like 50, 60, 70 or anything like that because of this. Because so their expression level of this lactase does not go down, still maintain high. And that is actually mutation. Okay? And a lot of those Western people, the white people, have this mutation. So, do you think this mutation will be the dominant or recessive? Yeah, dominant. Because you obtain a new function at the age of 40, for example. You're not supposed to have any function of this lactase. And then all of a sudden, as a consequence of the mutation, you get this function. So it will override your programmed normal non-function in this occasion. So this is dominant mutation. And this is actually, so it is a dominant mutation and it is also a point mutation. And another very interesting thing is, this point mutation is not on the actually coding region of this lactase, but is something far away from, so here, this is the area, on chromosome, this is the area where lactase gene is at, actually protein coding region. But actually that point mutation, C to T conversion, change, substitution, occurred at 14,000 nucleotide upstream. Can you imagine 14,000 nucleotide back? And this point mutation was something that causing this thing. So what is the problem? So that's why we guess that it is probably, this is far away from the actual gene. So what can that be? So that's why we guess that this gotta be on some type of enhancer. What is the enhancer? Remember still, enhancer is the area, the sequence element to which what? The transcription activated protein binds something. So because of that, probably some gene expression program has been altered, some activated, unusually activated. So that's how they can maintain still this high level at old age is probable, reasonable explanation for this malfunction. So in white Caucasian population is so prevalent, that also tells that all our human actually origin is something, we were all derived from very small number of individual population in the beginning, in the first place. So relatively these Asians and the Native Americans are extremely low in this mutation too. What about the Mongolians? They are also Asians, but they don't have any problem in this dairy product. So somehow did they overall pick up this mutation, do you think? No, it turns out that they don't have this mutation. But even with that, they have no problem of consuming this dairy product. What's going on to them? Microorganisms. Bacteria living inside of their guts. Somehow they harbor, they have different kinds of this microorganisms, so they will do this job. Okay, so today I don't have any time to go over this your student questions, so maybe next week, on Wednesday, I will have more time with your question. So today's lecture, I will just stop over here and see you on Wednesday.